Have you ever tried to find a single typo in a library filled with thousands of books? That's kind of what genetic research can feel like. So in this explainer, we're going to explore targeted sequencing, a really cool technique that's basically a search function for our DNA. It lets us zoom right in on the exact genetic information we need. So here's what we'll cover. First, we'll talk about the huge data challenge we're up against. Then we'll get into the solution, targeted sequencing. We'll break down how it works and what makes for really high-quality results. After that, we'll see its impact in the real world. And finally, we'll look at how this amazing technology gets into the hands of researchers. All right, so before we jump into the solution, let's take a second to really appreciate the problem itself. It's not just about data. It's about the absolutely mind-boggling scale of our genetic code. Okay, get this. The human genome has about 3 billion DNA letters. Now imagine trying to read every single one of those for every person in a study. It would be a monumental task, not just in time, but in cost. It's a true big data problem. And that brings up the big question for modern genomics, right? How in the world do you find what you're looking for in all of that information and do it efficiently and without breaking the bank? We need a way to just zero in on the parts of the genome that actually matter for our research. And that right there brings us to the solution. Targeted sequencing. This is the focused approach that lets researchers sidestep the massive challenges of cost and scale that come with looking at the whole genome. So what is it exactly? Well, at its core, targeted sequencing is a way to isolate and read only specific parts, or loci, of a genome. This could mean focusing on just the protein-coding genes, that's an approach called whole exome sequencing, or even creating custom panels to investigate a specific set of genes you think are linked to a disease or a certain trait. You know, this technology really does stand on the shoulders of giants. It all kicked off back in 1977 with the very first sequencer. Then, in '88, Multiplex PCR came along, giving us the power to copy multiple DNA targets at once. But a huge breakthrough happened in 2005 with a technique called hybridization capture, which really paved the way for the modern targeted methods we use today. This table just shows the trade-offs perfectly. Instead of trying to read the entire 3 billion base genome at a pretty shallow depth, targeted methods focus on much smaller regions. And because of that focus, we can sequence those specific areas hundreds or even thousands of times over. This is absolutely crucial for spotting rare genetic variations. The result? Lower costs and much simpler data to analyze. Okay, so we know why it's so useful. But how does it actually work? Let's dive into the two main ways it's done. The two big players here are hybridization capture and multiplex PCR. You can think of it like this. One method uses probes as a kind of molecular bait to fish out the DNA sequences you want, while the other uses primers to make tons and tons of copies of just your target regions. All right, with hybridization capture, you start by creating this molecular bait, which we call probes. These are just tiny pieces of DNA that perfectly match the genetic regions you want to study. You mix these probes with your sample, and they find and bind only to their matching DNA targets. Then, you add tiny magnetic beads that fish out the bait, pulling your target DNA right along with it. Everything else just gets washed away, leaving you with a pure sample of the DNA you actually care about. Pretty neat, huh? The other method is multiplex PCR. So instead of fishing, think of this one as a molecular copy machine. It uses a process that makes millions and millions of copies of just your target DNA sequences all at the same time. These copies, which we call amplicons, are then prepped for the sequencer. This basically drowns out all the noise from the rest of the genome. But, just getting the sequence data isn't the whole story. The quality of that data is what really, truly matters. So how do we measure success, and what kinds of things can get in the way? We use two key metrics to see how well we did. First is target region coverage. That tells you what percentage of your intended targets you actually found. Second is capture efficiency, which measures how much of your sequencing data is your target versus just random off-target noise. You want high numbers for both. That means you had a great experiment. Now, a few things can mess with these quality metrics. Some parts of our DNA are just chemically harder to capture. The quality of the DNA sample you start with is absolutely critical. And the genome itself has these tricky spots, like pseudogenes, which are kind of like old, broken copies of real genes that can confuse the whole process. Even long, repetitive DNA sequences can throw a wrench in the works. So, given how powerful and flexible it is, it's really no surprise that targeted sequencing has become this indispensable tool across a ton of different research fields. Let's look at some of its key applications. And the applications are just incredibly diverse. I mean, in human health, it's essential for disease research, like finding the genetic roots of cancer or inherited disorders. In agriculture, it helps breeders create more resilient crops. It even lets us push the boundaries of science itself, allowing us to study epigenetics, that's the layer of control on top of our genes, and even trace the evolutionary history of different species. So, with such a powerful and complex technology, how do researchers actually use it? Well, many turn to specialized services that can handle the entire process, from designing the experiment all the way to analyzing the data. So let's look at one such service from Genomics as an example of how this all comes together. The core value here is simple, but it is so powerful. By focusing all of that sequencing power on just the regions of interest, you can find rare genetic variants with incredible accuracy. And that's something you often miss when you spread your resources thin across the entire genome. For a researcher, the advantages are just crystal clear. You can find variants that are present in only a tiny fraction of cells. The data you get back is smaller and way easier to work with. The cost is significantly lower, which makes large-scale studies actually possible. And you get this incredible sequencing depth, giving you really high confidence in your results. A complete service like this handles everything, from start to finish. It all begins with a consultation to design the perfect experiment. Then comes all the lab work, capturing the target DNA and preparing it. After the high-throughput sequencing, the final step is a custom data analysis, really designed to answer the researcher's specific questions. And what you get at the end is just as important as the process. Researchers get the raw data, of course, but also a full experimental report and a detailed analysis. The service can even provide ready-to-use text for the methods section of a scientific paper, which helps speed up the path to publication. So this leaves us with one last really compelling question. With the power to go deeper and look closer than ever before, what critical, rare variants and low-frequency alleles, the very mutations that could unlock brand new discoveries, might be hiding in your specific regions of interest?